Okay, thank you. It's hard to follow such a great panel. My name is Betsy Norton. I'm an associate professor at Tulane University. And today I'm going to talk about some of our vaccination work. Um, and really, I want to start by just telling you who I am. My goal is to bring the combination of novel adjuvants to harness delivery routes for vaccination in my transformative research area. And so the way that we do this is we use adjuvants like DMLT or LTA1. If you're interested in them, the NIH has a nice compendium of articles that have been published. So you can use these QR codes now or later. And what's fascinating about these agents is that they help vaccines by allowing you to really harness the power of different routes around the body. So things like intranasal, oral, intradermal, transcutaneous, those aren't natural, I would say, consistent routes for vaccination. But what's amazing is that you get a lot of mucosal immunity from using these routes, which makes it a better vaccine. So we can do this with different types of antigen forms. And like I said, this is an advancement. I feel an advancement over traditional vaccine techniques because you can see these are classic vaccines that are given today, and they target a number of different diseases, a number of different antigen forms. The biggest thing that they induce is serum IgG. So what you see is that these injected vaccines, it's all about antibodies. What you're really missing is this mucosal component. This is very highly variable in these vaccines, and also T cells. And this means when you approach something like pneumonia, which has happened with COVID vaccines or even influenza vaccines, you're not getting protection at the site of disease. So you have issues with breakthrough infection, which really isn't quite breakthrough infection because those intramuscular vaccines are never going to be able to provide this type of protection actually in the lung. So the big, I would say, approach that we're taking is to actually try to create that protection in the lung with intranasal vaccination. And specifically, I'm talking about our ClayVax vaccine. This is the brainchild of me and Jay Coles, pictured here in our Tulane PR photo. And don't look closely at my nails because they're a little ragged. I need to do something about that. So this partnership is really about bringing LTA1, which is our novel adjuvant that works nasally, and Jay's outer membrane protein, which is a novel antigen that promotes protection to all Klebsiella strains, and also cross-protection to other Enterobacteriaceae. So the solution, as I said, is ClayVax, but what's the problem? So the problem is Klebsiella pneumonia, and this is a leading cause of bacterial pneumonia. It's the third leading cause of bloodstream infections, highest incidence of serious infections, and second leading cause of bloodstream infections with the highest incidence of serious infections. And really it's because there's this growing and major concern over antibiotic resistance and including untreatable infections. So some of our data, we've done extensive research. A lot of this is published in Scientific Immunology 2021 to show that the combination of LTA1 with this outer membrane protein of J's really reduces the bacterial burden in both the lung and the movement of bacteria into the spleen in vaccinated animals who have been challenged. This is inducing a T cell response as well as antibodies. And more importantly, it doesn't rely on what other vaccines will do, like capsular polysaccharides. It's providing a protein-based immunogen that's broadly reactive with all Klebsiella strains. In fact, we've tested 71 strains and they all have this antigen. And this is the challenge with a different capsule polysaccharide containing strain, and you can see complete protection with our vaccine. So it's a little bit different than, for example, the strep pneumococcal vaccines that rely on those polysaccharides. And like I said, there's a global concern, both in pretty much every country, about the growing antibiotic resistance. This is from an article in the Times of India talking about 30 different hospital centers and how much antibiotic resistance, Klebsiella pneumonia, in addition to other Enterobacteriaceae. This is the World Health Organization priority pathogens list for trying to figure out new antibiotics to treat these infections. Enterobacteriaceae is the family for Klebsiella pneumonia. It's estimated in some countries, half of the Klebsiella pneumonia infections are not treatable. And this is just growing. So if 6 out of 10 infections were treatable 3 years ago, now it's only 4 out of 10. So the vaccine, as we've shown in our publication, also this protein is highly similar among Enterobacteriaceae family members. And the T cells that are induced by this vaccination not only respond to Klebsiella pneumonia, but they actually have crossover to E. coli, Proteus, Enterobacteriaceae, Serratia. So we call it ClayVax because it has the potential to protect against more than just this one bacteria, but this provides some protection to this entire family of antibiotic resistant bacteria that are causing these problems. So the current data package, we've done safety studies with the adjuvant before we ever got into kind of moving forward with a vaccine that is published. We have a non-human primate and a rabbit study that's ongoing. I'll tell you, our rabbits handled their 2 doses just fine. Our adjuvanticity studies have been published, both the adjuvant alone and also the mouse, which is some of the data I've showed you. We have a non-human primate and rabbit study ongoing. Humans are planned in late 2025 and 2026. Efficacy, similar. And manufacturing, we have contract funding through the NIH for production, which we'll do at Rare. So this is where we are in our timeline. We have done our preclinical. We're kind of in our IND enabling studies, and this is our ongoing milestones that we have contract funding through NIH to complete. This goes all the way to a phase 1. And what we're looking for is partners once we get through this phase 1 into our phase 2 and phase 3. So our IP was filed, the provisional patent was filed in 2022. And as I said, we're interested in collaborations for this specific vaccine, but also spin up opportunities for using this technology in different ways. I don't just tackle pneumonia, I also tackle gastrointestinal disease with other novel combinations. And thank you for your time. I'll take any questions. Not a single. Okay.
Oh, here we go.
When was your provisional? What month was your provisional filed, and when do you plan to file for the full patent?
Ooh, I'll have to get back to you on that.
Where's Matt when we need him?
Yeah, yeah, yeah. If you ask me a mouse question, I'd be better poised to answer that one. But I know I've been seeing some emails about the patent, so it's probably, I think it was in the fall, and so it'll be coming up right now. Any other questions?
So do you see the cross protection across different species correlate with the degree of similarity in the structure of the OMP?
So we've only done it for T cell responses reacting to those antigens, and we have challenge studies ongoing right now to look at exactly that.
Hi there. I would like to understand, how do you see the space of resistance to antibiotics? I mean, there's a lot of approaches. What makes your approach unique?
Well, I think we're not in the creation of a new antibiotic space. So that's one. We're on the therapy side. So, sorry, the prophylactic therapy. And I think what makes this unique, number one, it doesn't rely on capsular polysaccharide, which means when you manufacture, you're not making, up to 12 or 23 different antigens. It also has the potential to be cross protective between strains, which is very attractive, I think, in a vaccination approach. And then, one of the easiest targets that we could see is the people, at least in the US, who are really at high risk when they have a pre-scheduled immunosuppression coming on, like organ transplant patients. Those are some of the most common infections that occur. And so they would be a great target for this. And those are also the people where you're not going to have the antibiotics working very well. Their immune system's already compromised. So I think it's a completely different approach. And our main competition would be other people trying to make this vaccine. There's one clinical trial that's ongoing, but as I said, it's more the polysaccharide approach versus the protein and a nasal vaccine.
Question right here.
Did that answer your question?
Yeah, so really interesting technology. Quick question is the durability response that you've looked at before. You alluded to the fact that it might be a prophylactic just now. So like, have you looked at how long the IgA, IgG kind of,
Yeah.
simulation stays?
So we have some protection out at least to 6 months, but we're in the process of trying to do that for a lot longer to make sure. It's possible that all the studies I showed you was a prime boost approach. It's possible that we would need, another boost, or in particular patients to have kind of, I wouldn't say annual, but maybe every 10 years to have a boost. We'll have to see how those studies go.